It's my great pleasure today to introduce the three winners of the Kavli Neuroscience Prize for 2014, each of whom has made an enormous contribution to our understanding of how the brain works, particularly with regards to memory and cognition. And three winners are sitting with me here: John O'Keefe, Brenda Milner, and Marcus Rakel. Now, I want to start with you, Brenda. Your career has spanned some 6 decades.
Yes.
And started in the 1950s working with one of the most significant contributions, a man who has contributed such a significant amount to our understanding of the brain, and known only by the initials HM. Could you tell us who HM was?
We can actually give his name now, because he died 4 years ago, and now it's Henry Molaison, and he came originally from the southern United States, but grew up in Hartford, Connecticut. That's where I met him.
And you met him in what context? Why was he in front of you?
Well, I met him because I had observed cases of memory impairment in Montreal. I was working at the Neurological Institute, as I still am, with Wilder Penfield's patients who were operated on for epilepsy. And we had 2 cases of severe memory impairment after unilateral, different operations from HMs. And we reported those at the American Neurological Association. So this was before HM, you know, and but Dr. Scoville, who was a surgeon in Hartford, was also at that meeting and read the abstracts, and he called Dr. Penfield and he said, I think that the memory impairment, the kind of memory impairment that you and Dr. Milner are describing is what I have seen in a patient in whom I did my operation. And his operation was different because it was in both hemispheres of the brain. All the Montreal ones were unilateral, but it was just confined to the medial structures, the deep structures of the temporal lobes, including the hippocampus. And he said, so I would like to invite Dr. Milner, I was in Montreal, to come to Hartford to study this patient and any other of my patients who may interest her. And Dr. Penfield said to me, would you like to go? And I said, you bet I'd like to go. And that was the beginning of how people don't often wonder about this, but I was a youngish woman, I was only 34 or something, from Montreal, be invited to go down to Hartford. Dr. Scoville was associated with Yale University. There were lots of psychologists. It was because of the paper that Penfield and I were publishing.
And what did HM, what was it, the characteristics that made him interesting following on from the surgery?
Well, of course, it's what is interesting in these patients is the preserved general intelligence of, you see, we're used to people losing their memories, unfortunately, many of us, as they get older, or maybe they lose their memory for something else, but then they go on living and they get further damage in their brain. So that when you, and this is long before any sort of imaging of brains, so any clues we were getting before these surgical cases would be based on pathology studies of somebody who died years later and had lots of other things wrong with their brain. And so how could you pinpoint exactly what was responsible for this gradual onset of memory trouble? Whereas when you have youngish people, HM in his 20s, our patients in their 40s, suddenly after a known surgical intervention, they were with normal memories beforehand, and then bang, right after the surgery, they have this amazing change with preserved good general intelligence, because if they were just a mess, a disaster, that would not be good. But it's the very good preserved general intelligence and ability to pay attention in a normal way, and sometimes better than normal because they're highly motivated, and yet forgetting as soon as you're distracted, and life is distracting us all the time. So as soon as you change your thought to something else, or your focus of attention to something else, you lose what happened before. And it's this continuous forgetting of what you had processed appropriately initially. So it's challenging, it's fascinating.
So what did he and your other work in this area of the brain, the hippocampus, tell us about what these brain structures were actually doing?
Well, it really tells you by default, it tells you what they're not doing. It tells you, you look at what is preserved and you see it's not into, these patients can, their normal emotional relation with people, socially very, HM was a delightful person. We all loved him. An interesting thing about HM is that when he died, we all felt we'd lost a friend. And you think of friendship as reciprocal, and yet HM never recognized me, recognized the people he was seeing, yet we felt he was a friend because he was so cooperative and so nice. It's a strange thing.
Every time you'd see him over the many years that you did study him and his brain, he never recognized you from one time to the next?
No. He was always very polite because he was naturally a polite person. But he wanted to help science. He grasped the fact that he was going to, be helpful, and just a few things over the years stuck in him. He knew there had been a president called Kennedy, and he knew that something bad had happened to him, and he knew something about astronauts. That fascinated him. He spent all his time watching television, and of course he didn't remember things from it, but he got these ideas gradually about space and so on. Something gradually built up, but that was all.
It's kind of tragic, isn't it?
Oh, it is. It is.
That you could spend so much time with someone and them not know who you are from time to time.
Well, that's an instance. Everything in such a life is tragic because all his dealings with people. But it's tragic, yes.
John, if I could just turn to you. You worked and continue to work on the hippocampus, which is the same area that HM had this lesion.
Yes.
How did you end up in the hippocampus?
Oh, that's very simple. I was fortunate enough to go to graduate school in the psychology department at McGill. And of course, one of the great influences at McGill, one of the stars already in the 1960s when I went there, was Brenda Milner. And I remember very, very vividly the first time I sat in on one of her lectures and she described HM and his memory deficit. I thought, wow, that is fantastic. I didn't work on the hippocampus while I was at McGill, but when I got the chance later on, after I had moved to University College, I thought this is an area in the brain which I think would really pay back and be rewarding to study. So we looked at the cells in the hippocampus of the rat. And I set out with the hypothesis that it was some sort of memory system and that I would find memory cells.
Was this based on, amongst others, the Brenda's studies of
Absolutely,
HM?
absolutely. And it was already proving difficult to find animal models of the types of deficits that were seen in HM. But I tried simple learning tasks such as training the animal to approach a particular object or press a lever to get food and things like that. And nothing that I saw in the cellular activity in the hippocampus reflected the behavior that was going on. So I thought, okay, I'm going to go back to square one and make believe that I had never heard of HM and that I had never studied with Brenda. And so I just went back and started to look at what the animal was doing in its natural day-to-day behaviors. And it's eating and drinking and walking around and trying to find various places in the environment. And it slowly dawned on me it took a couple of months of observing and listening to the cells, that the cells weren't interested in what the animal was doing. They didn't seem to be obviously interested in his memory or anything like that. They were interested in where he was doing things. And so it dawned on me that maybe it was something about the location of the animal that was the important variable. And then one day I realized that it was probably that the cells were, I knew definitely, they were coding for the location of the animal in space. And that led us to the idea that it was a spatial memory. So we came up with the idea that it was specifically a memory for space in animals, which then became elaborated to take on other activities and other memories to become what we now think patients like HM have a deficit in, is episodic memory. The memories for what you did in a particular place at a particular time.
Now this is in the 70s and 80s, is it?
This was actually right at the beginning of the 70s,
okay.
1970.
And most of the studies of HM, they start in 1957 and then continue all through his life.
Well, I saw him in 55, but and our patients in Montreal, 51, yes.
So which really, I'm leading towards asking about your contribution to the way neuroscience, the way we study brains, has fundamentally changed as a result of developing ways of looking at it, which is something that you've done.
It has. The catalytic event, of course, was the invention of Xray CT, the CAT scan, whatever you wish to call it, by Godfrey Hounsfield, which he announced in, I think, 1973, something of that sort. And of course, it immediately revolutionized the practice of medicine, particularly as it relates to the brain at the time, because his machine, for various interesting historical reasons, was devoted initially to the brain. That's where he got his support. And it replaced techniques that were difficult to interpret, where you would inject a dye up the carotid artery of a patient and see if the vessels had been displaced, looking for a mass lesion, and that carried a risk, or putting air into the spinal canal and letting the bubble of air go up, and then the patient would be in a chair and you'd tip the patient around so the bubble would float around in different places in the brain.
It's incredibly crude, isn't it?
Not only incredibly crude, but it was very painful. And in that day and age, neurologists, there were very few neuroradiologists, so neurologists and neurosurgeons did these studies. So I was accustomed to doing them, as having been trained to do this sort of thing. So overnight, these things were no longer needed. You could use Xray CT. But Xray CT, of course, just gave you an anatomical view. It basically looked at the density of the tissue by passing X-ray beams through at different angles. But I'm a neurologist by training, and I had gone to St. Louis, to Washington University, because of a physicist by the name of Michael Terpagosian, who had this vision that these curious isotopes produced by a cyclotron, positron-emitting isotopes, might be useful in biology and medicine, and particularly that it would give us the opportunity to look at brain metabolism regionally and potentially in a human. But again, this was injecting stuff up carotid arteries, so you could only look at this in patients. But this was a curious group. I was a neurologist in a group of physicists, chemists, computer scientists, and the like. And their first reaction to Godfrey Hounsfield was, well, we can build a better CT scanner. And there was actually a movement to do this that gave way when the institute in which I was in just decided to buy one of these things and get out ahead of the game. And in the office next to me one day, a physicist by the name of Michael Phelps and a computer scientist were looking at this, feeling a bit down, and they said, wait a minute, maybe we could construct a scanner that would use these isotopes, these positron-emitting isotopes, and reconstruct how they're distributed in a tissue, and essentially duplicate what you do in a laboratory animal, where you would inject something that would go to a particular place in the brain. You would sacrifice the animal, remove the brain, and lay it on X-ray film. And what emerged here was you could do this electronically. You could essentially decapitate a human being electronically, painlessly, and safely. And so in vivo radiography emerged in the group. And of course, you couldn't just go to a manufacturer and order one of these machines. They were built in the lab. They were constructed outside my office. And of course, I had a ringside seat, but I was neither a physicist, the guy that could build this. But the trick was to figure out how to use these machines. And my background had been in the area of brain circulation and metabolism, so we spent the rest of the 70s figuring out how to, use a machine like this. And then we got to the beginning of the 80s, and of course there is this long history that both brain metabolism and brain circulation mirror the changes in function within the brain. This goes all the way back to the late 1800s. And so the idea was, well, maybe we could take advantage of that fact and begin to develop maps of how the brain worked. And so we spent a while figuring out the details of how to go about doing that. And in the course of this, we were joined by, I think, one of the really outstanding cognitive psychologists in the world, Michael Posner. So we had figured out how to take a picture of your brain and then ask you to do something, take another picture and subtract the two and look at what had changed. And we then figured out how to do this across many people. And cognitive psychologists, of course, had been doing this behaviorally for many years, in which very precisely they would look at different behaviors and compare and contrast them by subtracting one thing from another. And this fit like a glove on a hand in this, and we began to take seriously the whole issue of how you go about mapping the brain. So this got that agenda going up through the 80s. And this was all with PET, and MR came after that based on.
As you were talking just then, I've just realized that there is a very peculiar connection between the three of us, which is that if you look on my wrist, I've got a scar there from a PET scan that I had when I was an impoverished student for a study. And it was in your lab. It was in your lab.
I don't think I put the needle in your arm.
They had to do it in both wrists because the artery shriveled up halfway through.
Oh dear.
I think I got paid £40 for that.
Oh yeah.
So thank you. How did the development of these type of scanning technologies transform your work? Because until that point, you were doing what Marcus was saying, which was effectively quite crude.
Well, it meant that you could look at, the normal living brain in action. I mean, it was most exciting because up till then we've been very much dependent on the lesion method and in the animal studies on experimental lesions, as you were doing, and with human patients, the surgical situation in which a surgeon gives you a really good report, as Dr. Penfield did, and you know more or less what's been removed, and you make your arguments from there, from what you find. But to have a normal, healthy person, because that's what we really want to know about. We want to know about the normal brain, and that you could have a normal, healthy person go into one of these scans, and you can give them tasks to perform while they're in the scanner and compare the patterns of activity and when they're doing one task as compared with the baseline task. Of course, it was all very challenging as finding new methodology, getting used to the limitations of the situation too, because you're not working with the free rein that you would have just bringing your patient into the lab. But tremendously exciting. But I would say particularly exciting that you could look at a healthy brain being active and learn something from it. That was really exciting.
And then, so moving on from PET scans, which are still used today, but in some ways is the technology been superseded by the development of MRIs and more recently fMRIs. Tell us about how that has transformed our understanding of the working brain.
We no longer had to do to others what we did to you.
I'm grateful for that.
It was an interesting bit of physiology that made this happen in part. All these stories have this wonderful history to them. Yeah. But the physiological history to the story emerged out of a discovery that we made quite by accident. The presumption is that if the blood flow to your brain increases when you do some kind of activity, obviously your brain must need more oxygen, maybe a bit more glucose to get the job done. And what came as a huge surprise, it was a shock to us and a bigger shock to the community, it wasn't well received when we first observed this and reported it, is that the blood flow actually goes up. So the supply goes up, but the demand doesn't follow it. And so the amount of oxygen in the blood in a particular area that's affected increases. Well, it goes way back to Linus Pauling in 1937, who noted that the magnetic properties of blood change depending upon how much oxygen is on the hemoglobin. And then there was a physicist at the Bell Labs was aware of our physiology, but also knew about Linus Pauling's observation. And he tested the idea that an MR scanner could detect this. And so all of a sudden, you didn't have to inject anything. The contrast agent was in there. It was nature's own, if you will. And so this really transformed things, most in terms of the convenience and safety of doing it. The infrastructure for a PET operation involves cyclotrons and chemists and so forth. Having an MR scanner, you can put one in the basement of this building and run it. You don't need this army of people. So this was transformative. And the other thing was that you could look at things in real time in a sense. In other words, you could design a behavioral experiment just as the psychologist had designed them in the first place, where you could mix different stimuli and so forth. That was not possible with PET. Everything had to be, you did one kind of thing and then you did another, and we made progress with that, but this was more elegant. So it became readily available, it expanded what could be done, and there was then, of course, a veritable explosion in what people were doing. I would say parenthetically that PET has remained a very, very valuable tool at the level of metabolism and a lot of other fundamental issues of how the brain works. We're still trying to understand why the brain plays these interesting tricks to allow us to look at it. And PET plays a very fundamental role in that. But just the mapping of the brain, which is a worldwide agenda now, is really been facilitated by MR scanners.
It's difficult to think of a more transformative technology in terms of how widespread it's been used across neuroscience globally.
It's really quite I've been almost taken aback by how much has been done, and you know, it's so available. And it's almost so easy to do that it is both, I think, been good at times, but then some of it, you know.
Well, I think some of it was very bad at the beginning. There was proliferation of bad papers.
Yeah, I think anything that becomes so readily available and is so easy to perform in one sense, but difficult to perform if you're going to, honest to God, do it right.
Right.
The literature becomes very complicated, but I think on balance that we've made real progress.
It's definitely a net gain. I know what you mean as well, that the reporting of studies of functional brains is sometimes what I would describe as suboptimal.
Yes.
I think that is a very polite way of putting it. You know, I've used harsher words at times.
John, I want to ask you about an experiment which is it's so dramatic to look at. It's so striking, and it's about understanding how mice and rats process where they're going. But it's the mechanical setup of this experiment is so beautiful. I'm referring to the one where you hold a mouse in place on top of a rotating globe, and they have a video projecting in front of them.
Yes. Yeah. So I should preface that by saying that we were also interested in using Marcus's fabulous techniques to see if we could actually demonstrate that the human hippocampus was involved in spatial navigation and spatial memory. And for many years we were sort of flummoxed, because most of the things that you can do with a patient in a scanner is you can give them very limited access to, you can ask them to remember words, or you can give them limited access to materials. And that wasn't the kind of thing we thought the hippocampus was doing. We thought the hippocampus was involved in actually exploring large environments and finding a way in large environments. And it was only in the mid-1990s that we realized that we could use virtual reality technologies to create worlds which are large enough for people to move around in and explore and find their way around in, and still do so in a scanner. And so I got together with a mathematical physicist, Neil Burgess, and he took one of the first games apart. He took one of the first virtual reality games apart, and he got rid of all of the politically incorrect aspects of it. It was Doom, wasn't it? It was Doom, and we went on to Duke Nukem and things like that. These games where you shoot up various villains and things like that. So he used his brilliant expertise to take the whole game apart and then put it back together so that it was just a very simple environment. And we showed that when you asked normal subjects to find their way around in such an environment, it actually only activated two parts of the brain, and one of them was the hippocampus, and one of them was the parietal cortex. So when, over the years, Neil would turn around to me and say, "Can't we do this on rodents? Can't we do this on animals? It would be really wonderful to get control over the whole environment." And I have to say, I said, no, they'll never understand it. But about 3 or 4 years ago, David Tank at Princeton actually showed that you could show a mouse a virtual environment in a situation where it sat there and it ran on a ball and watched the environment move past it. And it could actually act as though it were in a real environment. So we've been developing such a technique as well. And what this enables you to do is to completely control the environment, because you have control over all of the stimuli that are coming into the animal, and you also know exactly how the animal is moving in the environment. So we think this is going to be one of the technologies of the future, where you couple some of the wonderful technologies that are coming on board now in terms of optical recording, where you can look at the activity of many hundreds of cells at the same time, or use electrical recording to do the same thing, and to couple that with virtual reality for rodents.
I think it's wonderful that both that experiment you've described and all of the scanning techniques, their basis is physics, video games. You know, it shows great fusion of so many different branches of science. I want to ask you, as we wrap up, I want to ask you what your inspiration, your drives have been over the years, Brenda.
Well, mine is curiosity, I think, and curiosity about behavior. You know, it's very good if what you find out turns out to be in any measure useful in the, you know, for people who have memory problems and so on. But what drives me, it's really basic science. It's curiosity. Of behavior. You know, you see a patient does something or acts in a certain way, and then you want to investigate it in a more systematic way. I don't know where that particular curiosity about behavior comes. I think I must have had it, just been born with it.
Marcus, what is the question that has driven you throughout your career?
You know, I think Brenda captures it. You know, there's just something very neat about problem solving. And the brain is just a heck of an organ to think about things like this. There's so much that we can learn, but you don't feel like you're up against a wall here. There are so many different techniques, and I would emphasize the importance of everything from the most basic things of cell biology and genetics through neurophysiology and the imaging, and how these need to be complementary. But I just think it's a real privilege to be able to sit in the arena where these kind of conversations are held, and you're looking at a brain and you're saying, by God, I can understand this just a bit better.
Okay, I'm going to ask you a fantasy question now, which is, I'm in charge of a limitless grant. Okay? What is the one experiment that you would do with no restrictions on funding or brilliant people to work with? John, let me ask you first.
Well, it's quite, it's an appropriate question in this
Because
milieu
you just
and
got the
at this
money?
time. because I was at the original meeting, which was organized by, amongst others, the Kavli Foundation and the Gatsby Foundation and the Allen Brain Institute at the Royal Society Kavli Institute. In which they brought together nanotechnologists and they brought together neuroscientists, and they just asked us the same question. And out of that came what is now the Brain Initiative. And the idea is that I think a lot of neuroscientists, if they were asked that question and they had the available funds, would say, what we would really like to do is to be able to record from huge numbers of brain cells in different brain areas, in an animal, perhaps in a virtual environment. And that, of course, is exactly what the Obama Brain Initiative will enable us to do if the money is coming forth.
Well, what about you two? What are the questions that still need to be answered?
Well, I agree with you that if you could do all this recording, it would be tremendous. It's They're the same questions. It's the level at which you're answering them that's changing, isn't it? I mean, we have the same questions I have the same questions about understanding brain and behavior, and I was always brought up, even back in the 30s, to think about behavior in terms of brain activity, though we knew so little about it.
Yes.
A lot of psychologists don't care about the brain, fewer now than in the past, but a lot say they can do very, very elegant experiments, and they're very good in their mathematics and their analysis, and they think that, of course, the brain's important, but it's not for them, you know. But I think if you start out wanting to understand how the brain does its job, so to speak, it's really more of the same in a way. I don't think that I have a sudden new question that.
Yeah. So I think the point I'm making is that these new technologies are a game changer. They will enable us to do experiments which we couldn't do before.
Yes.
And by the way, I would, going back to your earlier question to Brendan and Marcus, I'm also curiosity driven. I'm in this just to understand things, to understand how the brain works. But I don't think we should ignore the fact that a lot of our basic approaches to medical problems, to diseases and things, come out of that approach. I think that there's a very good argument to be made that you start off trying to understand a part of the body, a function or something, and then when you understand it enough, you begin to understand how you could solve some of these problems. So for example, I think that diseases like Alzheimer's disease will benefit tremendously from all of the work that we have done and our colleagues have done in terms of the basic understandings. And we're now beginning to develop animal models of Alzheimer's and see if we can understand the basic mechanisms of
I guess I would say it's a two-way street here, and just picking up on what you just said, John, and that is that obviously there's a deep need to understand basic issues. Take Alzheimer's, big-time problem, terrible problem, financially and for everything you can think of. But it presents some fascinating questions about the cell biology of the particular parts of the brain, a particular system that we discovered by accident is the target of this. It has very interesting metabolic properties. It has a constellation of interesting cells. Yes. So if you can both start from the bottom up and think about the cells themselves, but then you can use the disease, if you will, as an experiment of nature, which should not only be useful from a practical, humanistic point of view for people's well-being, but also as a stimulus. I think it works. This is a kind of a wonderful relationship where a disease itself is, from a pure basic science point of view, is stimulating enough. It benefits both the people ultimately that have the disease, but it'll move the science forward and guide the views of people, including myself, who are interested in how the normal brain works. And you can use that interface, I think, hugely profitably. And so if I had unlimited monies, it would be to somehow or other leverage the broadest possible conversations among levels of analysis, because there are levels, for example, in the case of certain metabolic problems in the brain or how it works, that, for example, are far better understood by the cancer biologists. The best main fuel of the brain is glucose. And yet, and it's a major focus in cancer biology because cancer cells shift over to using this in a particular way that was characterized by Otto Warburg in 1929. It's called the Warburg effect. I don't think that whoever developed the biology on which we live envisioned that they would give you the Warburg effect so you could get cancer, but rather that you use it for biosynthesis and in the brain, memory and learning. We desperately need to understand how this works, and the cancer biologists are way ahead. So I would snare a few of these people, get them in the lab with me, and we would have a conversation along with John O'Keefe and others who work with these single cells and figure out how do these cells work, why do they choose one way of doing something versus another, but knowing in the back of my mind that there is the possibility that this will give me a clue also about something as terrible as Alzheimer's disease. It's just a wonderful environment where you have all of these wonderful tools, levels of analysis, and to persuade people that we need to have the broadest possible conversations while maintaining the expertise that we individually have at all these different levels. And I don't know whether money solves that or just a good bit of public relations, to which I tip my hat to the Kavli and all the people that have pushed neuroscience forward in this way to make people aware of the potential here. I think it's an exciting time.
Well, it sounds like you've got your work cut out for you for the next few decades.
Yeah, you're right.
Thank you so much for your time, and congratulations on this fantastic prize for your work.
Thanks.
Thank you.
Very much.